Good morning. So we're starting our Team 11 meeting today, Monday, the 18th of May. And so I wanted to start first with Claudia Pace, who's going to help us with getting access to the electronic medical library. I know as you are involved in all these projects, you may need to research the literature. And what Claudia is going to do is she can help us gain access to the up-to-date electronic medical library. So Claudia, uh, so everybody here is a volunteer, um, international medical graduate, basically rotating through Larkin. Um, so how do we best coordinate for maybe a group of them to have access to up-to-date so they can actually do some literature searches and then share it with the rest? I think that might be the best way to do this. Uzam, what do you think?
Yeah, I guess that's a good idea.
So how many people would need to gain access, do you think? Could we have one per project? Like, for example, each of the people leading each project, Saurabh, how many projects do we have going on right now? I don't know if Saurabh is on the line. Maybe Saurabh hasn't joined yet. I think at least 5 or 6.
Yeah.
So the question is,
So Jack, for them to be able to have access to the up-to-date, they have to have a Larkin email. So if anybody does not have a Larkin email, I can forward them a form that they need to sign so that IT can assign them an email. Once they have the email, I'll send them the instructions, and they have to log in for the first time from a computer in the building. So if they're in Palm Springs, they would have to do from a Palm Springs computer. And if they're here in the South, then they would do it from a computer within the hospital.
So what we'll do is,
Good idea.
some of you are not here, so you can work with whoever you have in your team that is either a local medical student or a local resident, and they can log in for you and then get you the access that way. So what I'm going to do is, Claudia, can you put on the chat the best way to get a hold of you?
Okay, it's my
They can
email.
email you or, yeah. So anybody the team leaders that need access to the up-to-date electronic medical library system, which actually is a very complete system. All you need to do is email Claudia and she'll coordinate with you, as you know. So you need a Larkin email, so we'll get all that coordinated and worked out.
Okay, so I'm gonna send my email in the chat, and
Please.
then I will be sending you a form that you need to complete. As soon as you get the form back, I will try to get you an email ASAP and then send it back to you guys. And then you can have access, okay?
Okay.
Right. I put it in there.
Okay.
How do we see this?
It's on the chat. Just press on the chat, and it should be present on you should be able to find it on the chat. Just press chat.
Press where you see on the right-hand side, there's like little dots and it says more. If you press there, then you can press chat and you'll see it.
I see chat. Okay. Excellent, madam.
Okay. So I guess, do we have an order in which you guys want to discuss all the pending projects? And then I have a couple new ones that are coming up that for people that want to, I also post them on the Telegram group. Is everybody on this call on the Telegram group also? Anybody who's not?
I'm not on the Telegram group.
Okay,
I'm not either.
Okay, so whoever, Uzam, do you have the link for the Telegram group that you can also put on the chat?
I'll put it in the chat, Dr. Michelle.
Right. So this is the best way to communicate, because obviously this meeting is only once a a week. So the best way to communicate is through this WhatsApp group. I mean, Telegram group. Um, so please sign up for that, and then you'll get instructions. Like as we get new trials underway, you'll get instructions on how to sign up for those. So while we wait for some other people to join us, so the way that we've structured this is there was an original group that basically came up with projects that went into the website for the FDA for clinical trials, the clinicaltrials.gov website. And they looked at the 2,000 trials currently underway in the United States and around the world on COVID. And then they compared it to the current Larkin treatment protocol, and they decided which, based on that, they kind of determined which of the trials were trials that we could potentially sign up for. So actually, Saurabh, who just joined us, did that work, and he came up with a number of trials. And I'm gonna have him go over what the trials that we're working on right now are. But I mean, I know some of you may not be on one of those groups for those trials. Now that doesn't mean that you cannot take the initiative and actually go on online. And on that website, the website is on the Telegram group. All you have to do is click on that, and then you will be able to select other trials that that we that you can also participate in. So this is not clearly limited to these trials. When I get, for example, there's now a trial, we get approached by a company that does the UV, is proposing extracorporeal UV treatment. They basically radiate blood with UV. And they actually are submitting it to our IRB, and also they're submitting it to another IRB. And so they're going to look to us to file the IND with the FDA. So that could be a new group that forms. I encourage you that, if you're going to sign up for more than one group, to please, if you do that, please devote the time to do it, because if you don't have time to do one, you shouldn't be doing 5 or 6 or 7 because you're not going to be able to get it done. I mean, it's a lot of work, as you know. I want to also applaud the group. Over the weekend, we got 18 people filed their city forms, which is what's required to be a PI or a co-PI or be involved in a clinical trial. This course is offered for free through our medical school affiliate. At the very least, that should be something you get out of this experience, is that, to be able to get that certification. And hopefully it'll be more than that. It'll be actually putting a trial together that will find the treatment options for COVID. So that's what we're aiming for. Saurabh, I'm going to turn it over to you as far as maybe just go over the trials that we are currently working on, potential submission dates to the FDA, and if you're looking for members for that particular trial or not. If it's full, that's okay. We'll move on to others. I have a few to discuss later today, but go ahead, Saurabh.
Yeah, good. Will you be able to give me a minute to present myself?
Oh, sure. Yeah, Felix. Yeah, go ahead. Dr. Felix Rivera Mariani from Larkin University. I'm sorry. Go ahead.
So I'm Dr. Felix Rivera. I'm assistant professor at Larkin University. So I mainly work with immune and genetic signatures from immunological exposure. So although I'm pretty much booked throughout the few months due to the COVID-19 pandemic, due to a few proposals relating to the pandemic. So hopefully I can provide a little bit insight on whatever I can help on.
Thank you so much. So just for those of you who may not know, so Larkin University has a College of Biomedical Sciences that grants a master's degree in biomedical sciences. It also has a College of Pharmacy that grants a PharmD, a Doctor of Pharmacy degree. And then it also offers a PhD in clinical and translational research. And in the next 5 years or so, we're planning to start a College of Medicine. So Felix is based at that building. It's a separate non-for-profit that we set up that basically is purely educational. So thank you, Felix, for being here. Go ahead, Saurabh.
Okay, thank you so much. I would like to talk about some of the progress we've made this week, and everyone has been so helpful. We've been working on a few projects like quercetin and BCG, and also working on tocilizumab. We have began to give some of the other projects to other students. Some students decided to work on ozone, and then there's also a project going on Xofluza. So we're making significant progress. We're looking to submit the BCG IND for the FDA within this week, possibly tomorrow or Wednesday. And we're looking to go for weekly submissions. We're hoping that we can make progress in this regard with everyone's support. For BCG in particular, so many people have been helpful. Esteban and Madhusudan and so many other people, and they will all be able to share some of their highlights at today's 3 p.m. meeting. And for BCG, I would just want to emphasize a few points. We're looking to make a trial which compares the BCG vaccine to a zinc ionophore, and that'll allow us to see viral entry. We can compare BCG vaccine to elderberry extract, which is known to have a potent zinc ionophore effect. We're also looking to start a second BCG trial working with a recombinant vaccine. And we're going to make this a unique approach because there have not been so many recombinant vaccine trials yet. Most of them have looked with simple BCG strains like the Tice strain made by Merck. So we're hoping that we can involve everyone and we can make these projects a success. We also want to assess biomarkers. We want to use something called the Olink approach, which is a biomarker protein discovery tool. And the biomarkers that we assess will include things like ferritin, statin C, serum amyloid A, interleukin 6, interleukin 1, which are involved in the cytokine storm that's created by coronavirus. We're going to look at fibrinogen because there's a cascade of events that occur from the acute phase reactants, and that causes hypercoagulability, and that leads to DIC. So we want to actually look at which markers are elevated in patients who have DIC versus non-DIC, who will have elevated markers in mild versus moderate versus severe COVID. Depending on the size of the study, if we have enough people enrolled, we may be able to look further into ventilated versus non-ventilated, maybe even into high PEEP versus non-high PEEP, and identify what markers are elevated in these particular patient categories. That way we can create some cutoff values which will better assist us in future trials. And our study will also include children. Typically, BCG studies have only looked at healthcare workers, and it's important to include children because if possible, if our trial is large enough, if there's enough enrollment, we may be able to find a few children who have Kawasaki's like presentations, because there's been a recent association with COVID and Kawasaki in New York. Granted, that's a much larger population, but there is a possibility we can catch 1 or 2 children with Kawasaki's like symptoms with antibodies against COVID, and we'll be able to detect that. As I said, we're looking to do the first BCG study with the Tice strain from Merck. We have to still contact a few other institutional pharmaceutical companies and create a sponsorship program so that the drug can properly be funded for our trial. So we will also look at recombinant products, and we're going to do that for the next BCG trial. And in particular, we want to use a nasal administration of the recombinant vaccine. That will allow us to focus on mucosal immunity. And the benefit of focusing on mucosal immunity is that you will be able to preemptively create increased IgA levels. And this will be more beneficial than an intramuscular approach. With an intramuscular approach, you get more IgG, and that's not a bad thing. But the problem is it's slow. So there have been studies previously on intranasal versus intramuscular BCG for TB. And in those studies, it showed that there's more efficacy and a quicker onset of activity for using the intranasal approach. So we hope to acquire a sponsorship from a pharmaceutical company for recombinant intranasal vaccine, and that will allow us to assess better which administrative method is best. We're also going to include prior patients who were administered the BCG vaccine before 1 year, before 15 years. These will be two different categories in our study. That way, along with a current interventional approach, we will also have a retrospective outlook in our study. And that way we can compare to see if there's any dwindling of the protection provided by the BCG vaccine. If it was given a year ago, or if it was given more than 15 years ago, we will be able to properly randomize all of these groups. And we have an approach for that as well. So I hope that this is successful, and I'm really glad that we were able to finalize the first draft of the IND. We are just putting the finishing touches on it, and we're almost ready to go. So thank you so much.
So that one's going hopefully in the next few days. Do you want to go through the rest, or actually whoever the leaders are can discuss them if they have let's do like a couple of minutes, within 5 minutes, so then we can move on to new projects.
Sure. So right now, the quercetin project is being led by Jordan, and there's a resident who's also working with us, Tashi, and she's a local resident, and she will be the PI for the quercetin trial. In the quercetin trial, we're going to do 2 different studies. One will look at the incidence, and one will look at the severity and the benefit that the coronavirus vaccination provides in both. So Jordan will talk more about this at 3 p.m. And then
they're talking about using drugs for Kawasaki disease in children.
Sure. So Kawasaki disease is a rare occurrence which is shown to have an association with COVID. It's been seen in New York. It's not really confirmed yet because they haven't done a biomarker protein discovery approach. And if we can find a patient, probably a young patient, in the typical demographic for Kawasaki, less than 5 years old, then we will be able to identify those biomarkers, and that'll let us create some kind of a prognostic approach for this kind of patient. Does that answer the question, or do you have any other questions?
I do have one suggestion. So I would advise to keep in mind to expect small sample size, so single biomarker will not work. It has to be a shotgun approach. You have to identify signatures of biomarkers.
I agree with you, and that's why I was a little bit concerned that we wouldn't be able to do much on Kawasaki, but I think we will have a large enough sample size to compare mild versus moderate versus severe at least. That will allow us to identify the appropriate biomarker elevations in those patients. So we can at least do this much, for sure. If we have a large enough trial with multi-center involvement by sending investigational brochures to different medical centers and getting their IRB approval, then we can also see something about ventilated versus non-ventilated, DIC versus non-DIC, these kind of approaches. We can look into this a little bit, depending on the size of the study. Kawasaki is a little bit of a shoot for the moon, you know, if they are that many, depending on the population. So I don't think that would be the best approach to go for Kawasaki, but I do believe that that what you're saying has validity for sure. And depending on the size of the study, depending on the end, that will let us better assess our approach for this. I've worked with Dr. Patel, Dr. Madhusudan Patel, and he's also here joining us on this meeting. And I'd like to, you know, give the floor to him and have him share his opinions on BCG as well. So all yours, Dr. Patel.
Hi, everyone. Thanks, Saurabh, for introduction. My name is Madhusudan Patel, and I'm from Chicago. Glad to work with this exciting clinical trial. Let me just give you a little bit background so you can have idea, like before we discuss about the topic. So I'm an USMLE tutor, step 1 to step 3. So I've been in the knowledge of step 1. I've been tutoring since last 2 years. So Saurabh and we discussed certain aspect to look at the clinical trials. Dr. Michelle is here.
Yeah, I'm here. I just muted
Hi.
so you didn't hear the noise, but yes. How are you?
I'm good, sir. How are you?
Nice to meet you.
Nice to meet you. So he told me a little bit background that there are certain clinical trials that wants to run. So BCG trial, I think it's an amazing idea. Especially there are 3 concepts that he discussed about. One, actually compare with a natural component extract like an elderberry, which has good amount of zinc ionophore as a prophylactic. Second was BCG, but recombinant BCG, which is recombined with any antigenic envelope antigen with either influenza, which we compare with the flu shot, and compare
Any heterologous antigen.
Any heterologous antigen, not necessary it has to be influenza, it could be measles. There are different kind of recombinant BCGs available. But I was just putting that influenza because it's every year, it's a very common flu shots that every patient receives. So when it comes to patients, understanding and receiving that medication, they wouldn't have problem. Other thing, third one, BCG, we are going to compare with just as a laboratory reference changes. So acuity and the severity of COVID patients actually varies. It depends on how much immunogenic response it creates. So if they are well-trained macrophages in primary defense mechanism, that's what BCG is aimed to do. Our alveolar macrophage to be more trained as epigenetic changes and all. So I think it is a good idea that Saurabh actually described, maybe yesterday spoke, and he said that we can correlate acute phase reactant profile, especially in severe patients, either they have DIC or non-DIC group, and seeing after BCG vaccinations, how these after 3 months, how it changed cytokine performance and acute phase reactant level. So these are the 3 concepts we discussed yesterday. But there is another concept that today I was thinking it is just we can discuss. First, there is a recombinant BCG has been done before, even with measles, influenza, and they have tried in mice, and they have shown some good good approval in fighting respiratory infections. Now, that passed to SARS-CoV, which generally in 2002 in China, and MERS in 2012, which was another strain. Current coronavirus has certain genetical sequence and certain genetical information very similar to MERS. So what I was thinking, it will be a good idea to find a company, because recently there was a California biotech company which recently found an antibody which can bind to an outer envelope of coronavirus. So finding a similar antigen which is present in the outer envelope, which can generally create an antigenicity, not necessary, it doesn't have to be alive, because there is a company, if we can find a way to recombine BCG with just an epitope of an antigen, I think it will be a good trial that nobody has. I think so far they haven't done it, any recombined vaccine.
And this would be something that we would need some type of manufacturer to do, right?
Yes. So there is a company called the California biotech company recently found an antibody which is generally targeting against outer envelope, right? So this kind of company, it's called Seranto Therapeutics of California biotech. If they can biogenetically engineer, BCG, recombine with some of the epitopes of coronavirus, whichever is, if not current coronavirus, because people already have an anxiety inside, not receiving anything which has a corona particle, but the previous, like MERS or SARS-CoV-1, coronavirus 1, that generally whatever are very similar epitopes, that's something biogenetically can be engineered. And why it will be more beneficial, the route that I was thinking, because I've been reading about BCG since last 1 week. What I have gathered the information and found out that when we give intranasally, the main aim is just to train innate lymphocytes, or our primary defense mechanism, as a barrier. We have to do epigenetic, transcriptional and metabolic changes and mature up in a nice way, so smaller or less lethal respiratory infection doesn't harm much. So it's like a training our macrophage natural killer cells and dendritic cells. So BCG does that independently, even going through adaptive immune response. So if we recombine, and intranasally, because IgA is what we are targeting as a added effect, even if it goes into adaptive response, I think IgA is, we should target more as the immunity, as the first barrier.
Kunal is pretty good at getting hold of companies. Kunal, you're on the line.
Yes, I'm listening. Hey, Dr. Patel.
Hi.
Hi. So I actually sent an email to Saurabh and Dr. Michelle earlier today. So I'm a little late on catching up on BCG. I've only been doing reading since yesterday, so my apologies. But you can enlighten me on this. So I came across something where it said that Merck, the manufacturer that we're picking, they're not one of the WHO pre-qualified for BCG vaccine. So that pretty much means that there are concerns about the quality, safety, and efficacy of the BCG vaccine. So I was wondering, I'm certainly happy to bring up these questions to Merck people, but I was also going to look into if there is a possible way to collaborate with one of the 4 who are actually WHO qualified.
The thing is, it could be a game changing. There are not many trials which are done, but I understand. Thank you for, it's very important to, find safety, because it's a live assimilated vaccine.
Exactly.
Especially BCG, and it is not a normal protocol that everybody receives. It's not in a schedule, which I really understand compared to other countries. So yes, safety is a major concern, but this was just another topic. Maybe we just need some companies who can actually look into, and more immunologists, who can guide it in a right way, just to see if it is possible, just for new change, right? And rest of the things, BCG has been tested before in different countries, and right now the concept, very similar concept, is running in Pune in India. There is one trial. They already,
Yes, I saw by the Indian Serum Institute, correct?
Exactly. And they actually started this new, the same, whatever concept we are thinking, BCG, they give to healthcare workers or any high-risk population, right? And they see how effective it is not contracting coronavirus. It's just to train our primary defense in our lung. So, but there could be some errors and everything, because in India, it's in the schedule. Some people have already received BCG when they were a child. And if you look at it as a retrospective way, there is a decreased incidence of finding more coronavirus, knowing that that country is very dense with population. Even social distance, even if they try, doesn't work much.
Dr. Patel, Arpit Mehta here. So yeah, I have looked into the associations of BCG across the populations in all over the world, and all the countries which have them in the schedule actually have low prevalence. And there are some countries where it's it started, but it's not compulsory, and the compulsion date was later. So the older population which did not receive the vaccine has higher prevalence of corona as compared to the population which is younger and have received the BCG. So there's evidence which is stating that, it has been, if not like, curative, it is like preventive or lesser prevalence present for the BCG in all the populations where it has been administered at a part of the program.
The larger study came out from Israel, a country that, by the way, is doing very well with this crisis. Have over 72,000 people, and they didn't find evidence that BCG protects or changes the outcomes for these patients. This is an observational cohort. And
there could be many confounders, not sometimes confounders in study. If they don't include, there could be a lot of biases.
That's the major confounder when they find that association, because the countries with BCG effectiveness happens to be the countries that have the lowest amount of people that is older than 65 years of age. So I think before we go into this, I really want to see a very good, strong review of the literature with evidence in favor and evidence against. Because also you have to see if it's indicated for your specific population. So I don't want to come into conclusions because I don't know. The concept is fascinating, by the way.
Thank you.
But even if you're going to have this contact with a pharmaceutical company, they're going to ask for a rationale. And that rationale has to be supported with not only a concept, which mechanistic concept that is okay, but also you need to have some, studies that support that idea.
I can look into that, all those studies, first, by looking into clinical trials. These topics, we can first filter if there are very similar studies are even done with the larger population level, knowing the clinical statistical significance. So that generally, and then after I'll find all those information, I'll combine in a file. Okay, whatever recombinant BCGs, how they were in mice and in human, and I can make link all of those, abstracts, and I can send it to you.
I honestly don't see the harm in Kinal trying to reach out to some manufacturers and just give a preliminary expression of interest, because they might just give resources. They might assign someone to it. So if you want to do that also while you're researching, I think that's a good idea.
That's
I'll be
why
on it. Yep, sorry.
Go ahead. Sorry.
And you don't have to be bound with Merck, by the way. I mean, if you find a manufacturer across the globe that will work easier with us, because sometimes the U.S. companies just they don't want to work. They're not interested in. So you might have an easier time especially we have people here that are in India right now that can possibly make the connection.
Yeah, and similar to what Dr. Marcos was saying, a lot of the trials are targeting the high risk population. So they're elderly, they have comorbidities, as well as they are, they're somebody who come from countries where TB is highly prevalent. And so a lot of the ongoing studies I looked at for BCG were also designed in a way we're only targeting like healthcare workers, obviously because they're at high risk. But yeah, I agree that I could certainly start reaching out to different pharmas and just be upfront and honest about questions and concerns we have about manufacturing side while we are still doing our literature review. I think it's worth pausing for just at least maybe a few days so that we can look at both sides of the issue.
Sounds great.
It doesn't matter where you send this, because I've done this many times. If you're gonna get ECN, they're gonna say yes, but then you have to contact scientific medical liaisons in the medical team, and the best way to do it is a one-pager rationale. There's no other way. And you can get the product basically for free, and sometimes they can even make a match placebo, because we haven't done it with different nutraceuticals and components like that. I did trials for L-citrulline. I did one for omega-3 fatty acids. All of those I got, basically the components and placebo. But they want to see a rationale, and it doesn't have to be 10 pages. It could be a one solid page with all these little components.
What Madhu, you just described is a rationale. I mean, I think it's probably, you probably have enough to put something together. It's a theory. You don't have to prove your theory. So
it's in a concept which can be further if there is a proper support, it can turn into a new vaccine protocol, which can help. But we have to, yes, I agree with you, Dr. Sanchez, that we need a solid, a paper also saying, proving that whatever, concept that we are presenting. So I look into those, all those pros and cons and what real studies are done. BCG incorporated with other recombined heterologous antigen or epitope, and how was, and the result. I'll send you the link.
I would like to elaborate a little bit further on Dr. Sanchez. And we have to be cautious to not be too ambitious on what we want to try to accomplish with those trials. Once it is noted that it's too ambitious, it's not going to go anywhere. And again, as I mentioned before, we mentioned comorbidities. One of the strategies to overcome the comorbidities in an experimental approach is to look at biomarker signatures, not single, not 10, not 5, 10, have to be a bunch of biomarkers. Because that allows, even if you don't find statistical power, it will provide an estimate that you can support the finding. So we have to keep in mind that we cannot fall in love with p-values. P-value doesn't mean nothing. The effect size, if you have effect size, even if the statistical value is low, it is as significant as having a good p-value.
It's probably doesn't even have, you just have to sign it. He's got a standard form.
Saurabh, do you
They can't send you a.
the BCG, do we have, who's in charge of that? Who's leading it?
So I think Saurabh has left the meeting.
He's right here.
I'm in the meeting. I'm right here. Hello?
Oh, okay, you're there. Okay, sorry. I heard that, I'm going now. That's why I saw the message. Okay. All right. Yes, Saurabh.
Is Madhu leading this team, or how do you have it structured? Do we have it in
So first, yesterday I just, I was just telling him about the frame that first, how many clinical trials that the topics, that needs to be discussed, the framework of the studies. So first I was telling the BCG can be compared either with the natural component extract as a zinc ionophore, which we are planning to do elderberry, and also try to compare with recombine with another vaccine. That's another type of BCG. And third one will be BCG, just to see the lab value, if there are any changes. So these three components we discussed, and quercetin component, also, I was helping with another group. So we need a proper initial frame, so it can give us an idea that, like, how to approach. First, for that, that flow needs to be at least understood, that like what we are looking at. And that's what I explained. I discussed with Saurabh yesterday, and I think Saurabh will be basically Saurabh and I will be helping, for putting a frame in all these quercetin and BCG, right, Saurabh?
Yeah, of course. I can elaborate on anything anyone has any questions or suggestions.
So for this team, do you need any more? Do you need anybody else, or you're full?
So our thought was that we can go ahead and submit the IND that the team has created. We have about 25 people, and we can go ahead and submit that using the simple non-recombinant BCG vaccine, just a typical approach without comparing it to any other vaccine. A very simple trial, just only to look at the efficacy of decreasing incidence of COVID in patients who have BCG, and also checking the decrease of severity. And that's all. And that's the basic simple trial that's ready. The other things we're talking about,
the next couple of days.
Sorry, go ahead, sir.
That one's coming out in a couple of days.
That is ready in 2 days. That's the simple trial.
That's great.
That's good to go. We just need to make it become in a single pen. That's all. Because right now, because there were about 15 or 20 people who contributed to that, and they are exceptionally hard workers. They did a great job, but the problem is it's choppy in terms of the writing style. So I'm working on that, and we're going through the peer editing right now, and that will be ready to go in 2 days. And that is just a conservative trial, conservative approach to assess the efficacy of BCG in decreasing incidence and severity of COVID infections. That's all. Other things that we brought up or were considering, like including a recombinant vaccine, including a intranasal approach to allow for getting IgA activation with innate immunity. These kind of things we thought it will be better addressed in a separate trial, second BCG study. For that BCG study, yes, we do need more people.
Okay, so anybody that's listening that wants to participate can do that. Just reach out to Saurabh. Saurabh, who's your local champion for this, for the one that you're about to submit? Who's your local?
For the one that we're about to submit, what we need the most is a PI. We will need a principal investigator to be able to work locally at Miami, at Larkin, to be able to administer the BCG vaccinations to the patients who have given their consent. And the thing is, I'm actually, I'm not in Miami currently. I would love to be, but I'm currently not. And I believe that a local PI would be best. The way we have Tashi for quercetin, she had agreed to be the PI there. And I understand she's doing her residency and she's busy and has many tasks. I understand this is not easy for anyone to take on, but I would really appreciate if someone would be willing to work with us to administer the management of this first trial locally. And so I would be honored to work with any of you.
I spoke to Saurabh yesterday regarding this, and I told him like the situation from my side is going to be somehow complicated, especially the fact that our hospital is related to HCA, which have like a central IRB, which are very like.
I get it.
Yeah. So I told him, I think not to just to make things simple from your side. If we can find someone from Larkin, for example.
No, we definitely can. Marcus, who do you have on site that can do this? That the CITI trained?
Oh, virtually every resident, virtually any of them that they can be PIs. The thing is, the PI has to be in the institution. It has to be part of the institution.
Right, no, I get it, but who do we have that we can offer SORAB right now? Maybe anybody, any Larkin resident on the line that wants to do this, or should we bring it up at the 4 o'clock meeting?
I will bring it up in the 4 o'clock meeting because I'm not getting many volunteers stepping forward.
The 3 o'clock meeting, right?
Or 3 o'clock, okay, 3 o'clock. For plasma, we have the oncology group there and one internal medicine resident. So we need to do the same in each of these different, trials. See who wants to. I think was interested in quercetin at some point. I don't know if she's here in this phone call. She's rotating at this point.
Who are you talking about?
Family medicine resident.
Oh, I know Novera is on. Novera, are you on the line? I see Novera and Jeffrey. I don't know. It's muted. Or what about Adarsh? I mean, Adarsh, are you available for this?
Adarsh is working with me in the ozone, but
No, I don't want to move people back and forth. So just anyone else that's local in Miami that we can give to. If not, we'll talk about it later today.
One of my team leaders for quercetin, his name is Jordan. He's on the meeting right now. He works with many residents at Larkin, and I'm sure that he might be able to contact a few to be able to possibly work as the PI, and because he has many of his at Larkin.
Jordan, go ahead. Who do you know?
Hi guys. So I work at Design Neuroscience Center, and so we get a lot of the IM and PMR residents coming through. I would have to go through the list and see who's in the building, and also go through the list of which numbers I have to see if I can recruit anybody.
Okay. So that's the plan.
Yeah, as of now, I don't have any specific names.
Does the PI need to be done with PGY1 and step 3 to be able to administer the medications, or are there some other criteria?
It has to be a licensed physician.
Meaning they need to be done with step 3 and PGY1, right?
With Florida license too, yes.
Okay, just wanted to confirm.
For study coordinator could be anyone. For co-PI or sub-investigator could be anyone with the city certification. So the city certification comes into the part of the IRB application.
Okay.
So that's pretty much the requirement. So some of the volunteers, they can be sub-PIs, but I definitely need PIs. I'm running short of those.
Sorry, but you planning to be at the 4 o'clock meeting today? I mean, at 3 o'clock meeting today, or no?
Yes, sir. Of course, I'll be there.
So why don't we bring that up? Because all the residents are going to be present, and all the attendings are going to be present. So we'll figure out who.
Okay, because we're almost ready to go with this first IND for BCG, and I know we were working jointly on quercetin and tocilizumab, and I was also I'm also working to guide Kanal for her Xofluza project, and I've also been trying to set up a new project for plasmapheresis, and that's why my efforts were a little bit divided here, and that's why my target was actually to be ready to submit the IND today itself, but I think it'll take about 2 more days, and we also do need the PI before we can submit, because for the forms to be sent in, it requires PI and then co-investigators, sub-investigators. They have all this paperwork to fill out.
Yeah, we'll have that defined by the end of the day today.
Yes, sir. Perfect. Thank you so much.
Let's move on to other projects. Saurabh, do you have any?
I would like to discuss quercetin next, because quercetin is where we stand at completing as soon as probably maybe within the following week. We might be looking at sending in a submission for the IND. So Jordan will be discussing our current progress with quercetin. Thank you so much, Jordan, for all your efforts.
Go ahead, Jordan.
All right. Hey, sorry, I have a document here with talking points that I wanted to go over. I just got to find it real quick. Okay. All right. So the quercetin study, what we wanted to do is we wanted to show that quercetin could be a better option for prophylaxis and possibly even treatment than protease inhibitors such as lopinavir. Some of the reasons why we wanted to do that was because of not only the 3CL pro protease inhibition that quercetin has been found to accomplish, but also immune modulation, antithrombotic effects, and inhibition of viral entry into host cells, which has been documented in other peer-reviewed articles. We currently we're looking at using a metabolite, quercetin-3 glucoside, because of the bioavailability and it being taken up by the SGLT1 receptor. We're talking about whether or not we should be adding things like vitamins to increase bioavailability as well as discussing what kind of doses we want to use. There's another study going on right now using 500 milligrams of the quercetin in order to prophylactically manage patients who have not tested positive, and then 1,000 milligrams for patients who have tested positive. We were discussing the possibility of doing weight-based dosing as well. So we're the.
You want to share your screen? You want to share your screen?
Oh, no, that's okay. My screen is pretty disorganized. No, I'm just reading right now. So we've been looking at a couple manufacturers. We looked into Merck. We looked into a couple of other manufacturers. So far, we haven't had any luck in terms of somebody producing a tablet or a pill that we have been confident in. But we've seen a couple different names come up and are still investigating where we're going to get the product from, who our manufacturer would be.
So Jordan, this is Kanal. I just
Mhm.
had a couple of things to say. So I read one of the couple of sources. I'll have to look up again. But there were trials with quercetin where they used vitamin C and acetylcysteine as well as zinc. So it was kind of like a combination of the four different things. And then we also have someone by name of Dr. Sharma in our Team 11 group. He is like a renowned Ayurveda nutraceutical guy who also has a lot of research experience. So he may be able to help you or Saurabh with not just like the peer review for quercetin, but he may be able to guide us as far as like what manufacturers to trust, and like he's very knowledgeable. Like he's deeply involved with medical affairs as well.
What was his name?
Last name is Sharma, Sharma.
Okay. All right. Thank you. Can I get his contact information from you after
Absolutely.
the meeting?
Yep,
Okay.
absolutely.
Thank you.
Thank you.
Otherwise, we have a draft right now. We have a rough draft for a new drug application, and we currently have assigned our members have been assigned sections to complete, and we're going to be reviewing those throughout the week.
Excellent. Saurabh, you want to keep going?
Yes, sir, definitely. So after we complete the quercetin project, we will focus on working with tocilizumab. And where we stand with tocilizumab is tocilizumab is an interleukin-6 inhibitor, and we are hoping that it will help with the immune dysregulation that is commonly seen in COVID patients. So generally, COVID patients display a lymphopenia with elevated interleukin-6, and tocilizumab can work as an interleukin-6 blocker. And we will develop a study comparing a IV infusion of tocilizumab, probably 4 to 8 mg per kg, over, I think, 12 hours. And that will allow us to compare standard of care therapy to standard of care therapy plus tocilizumab. It'll be a very simple, conservative trial and very clean. No, it'll be quickly designed with our IND teams. We have the forms set. Actually, I've also shared the same forms with the new leader of the Ozone Group. And this is just a list of forms for 1571, 1572, and associated forms, which allow for submission of a investigational new drug proposal. So that is our approach for tocilizumab. So we hope to get that done as well after quercetin. And again, with quercetin, we're going to take it into 2 trials. So we will need, I am not sure if Tashi will be willing to PI for both of them, depending on her time constraints. So we may possibly need one more PI for quercetin. We will also need one PI for tocilizumab. So I would love to work with any of you who are interested in a PI for this study on quercetin, the second study on quercetin. Remember, one is looking at incidence of COVID if someone is administered prophylactic quercetin orally. The second is looking at the severity of COVID. So we possibly have a PI for both. It depends on her availability. And for tocilizumab, we definitely need a PI. And I look forward to working with any of you and learning from all your experience. So then after tocilizumab, we are working on plasmapheresis. So as you know, there is a approved trial on convalescent plasma at Larkin. Our approach is different. We're going to work on plasmapheresis, meaning plasma exchange therapy. So this is also known as PEX, P-E-X, and it has been shown to be a effective therapy for a number of autoimmune and rheumatic conditions, and these have shown to be tremendously benefited by plasmapheresis. So this is a known approach. It's nothing that is very unknown. It's been thoroughly studied. So plasmapheresis, the adverse effects are also analyzed, and we will need to submit a investigational new drug application for plasmapheresis, as you know. So we hope to assess what is the effect of therapeutic plasmapheresis versus combining plasmapheresis with Medrol. So there have been studies on plasmapheresis in COVID patients, but our approach will on understanding, should we combine plasmapheresis with Medrol or not? Because what is the point of plasmapheresis? Basically, we want to alleviate the cytokine storm, right? So Medrol will also calm down the immune hyperactivation. So basically, we want to know, should we take this plasmapheresis front in this war against COVID? Should we do a 2-pronged approach with Medrol and plasmapheresis, or should we only do plasmapheresis? And this is where the unique aspect of our study lies. So again, we will need a possibly, right now, Tashi is interested in being a PI for this. And if she can be a PI for this and quercetin, it's wonderful. And I look forward to working with her. And if anyone would like to be a co-PI, a sub-investigator, or just help us participate in the project, we would be glad to learn from you.
So we'll bring that up at the 3 o'clock meeting today. I'll just have you start with that so that the people on the call can sign up. I'm sure we're going to have a few that will. So
yes, sir, of course. And again, with plasmapheresis, what we hope to do is run a second trial. And the thing is, we can run this trial in a similar way to what we hope to do with BCG. So as I mentioned, we want to use a company called Olink to get the protein biomarker discovery tools. And we can apply those same tools that we do with BCG to plasmapheresis as well. And that'll let us evaluate the exact biomarkers which are elevated in mild, moderate, severe COVID patients who have been administered therapeutic plasma exchange. So that way we can create some kind of established protocol or guidelines of what biomarkers should be assessed before giving someone plasmapheresis therapy. So this is another, that will be our second plasmapheresis trial. So we are going to work on that as well. So basically first we have one current BCG trial ready to go. We're gonna do the second intranasal recombinant BCG vaccine trial. Then we will do the 2 trials on quercetin. Then we will do the 2 trials on plasmapheresis. And we also have the ongoing ozone trial, and we will also do a Xofluza trial. I hope to incorporate an Olink approach into Xofluza as well. That way we can possibly create a second trial with Xofluza to assess the biomarkers elevated in someone given Xofluza, which biomarkers are calming and which ones are elevating. It'll be important to assess this. Then after this, there are some other novel approaches which I've found in some trials. They're looking at some very common compounds, and we can easily acquire these. So they have strong antiviral properties. I looked carefully at their EC50s and their lethal dose, and they have a high lethal dose and a low EC50, meaning that they have a strong, broad therapeutic index. The one that is most appealing from the list of flavonoids is EGCG. And this is a green tea extract. It's catechinin. And we can perhaps work to administer EGCG in a dosage which will be at the right level to prevent viral entry. And we can also use a approach of comparing EGCG with other flavonoids like quercetin, and we can also involve compounds which are shown to have potent effects, natural products which are shown to have potent effects, and we can do a comparative trial of natural products against coronavirus.
Sorry. I actually agree with you. That is actually, Dr. Michel, I wanted to pick up new component. There are certain studies showing a promising COVID-19. You are saying a control group, there is astaxanthin. The recent study, I think in March 2020, they showed that it is a powerful antioxidant that generally we find in fish oil or krill oil. It generally when cold water fish, when they eat algae or phytoplankton from the sea, that's pigmented, the reddish color of salmon is astaxanthin. So there are extract available, and I think that natural extract has a lot of power, preventing certain entry of viruses and helping with proteases, inhibitor. So if you want to, I think quercetin and astaxanthin, maybe cross studies, will be a better idea.
I like that idea. I think if we do take after doing all those other studies on quercetin and plasmapheresis, if we can do a comparative study looking at astaxanthin plus standard of care versus EGCG versus EGCG with standard of care, and we can assess which one has a greater EC50, we can be able to incorporate some kind of protocol in administering phytochemicals and flavonoids, and that will be a great contribution.
I agree with you, and I because in quercetin, Jordan. Hello.
Yeah,
Jordan?
I'm here with you. Yep.
Yesterday, so far in a control group in quercetin, we are planning to. There was one with the zinc ionophore. Right. It just what control group we have added in study?
When we were talking about the zinc ionophore yesterday, I believe we were talking about the BCG vaccine.
What is the quercetin that we are comparing as the control group?
The isoquercetin.
That is glucoside forms, right?
Yeah,
the beta D-glucoside.
The quercetin 3 glucoside, exactly. Yeah.
So what I suggest, this is just an idea. If we, because people, there is a study done for any viral entry defense, like primary defense mechanism of respiratory with quercetin. And there are separate study done for astaxanthin. So I think it will be a better idea to cross this natural extract and create a study and to see which one is better, especially COVID-19 severity as a prophylaxis. Hello.
Hello.
Yes.
What do you think, Dr. Michel, about these cross studies with different extract?
I totally agree. I think, you know, there's nothing to keep us.
Hermano, máscara
So,
de pintar. Entre más,
I,
en ningún lado. No sabes dónde poder.
I totally agree. I don't see why we wouldn't do it. So I agree, Manu.
It's something different than what's already been studied.
That's the thing. It's sometimes people are get fixed on these studies and they don't, you know, want to try innovative ideas. And I'm all for it because, you know, you never know. You know, we might come across something that might make a big difference. So I'm totally behind.
Thank you for motivating, because this is a platform that, you know, all those, you know, ideas that we can present it to you. What we need maybe a little bit, there's a different study either we can create with quercetin or first I wanted to know how many trials, you know, that we are aiming for. So that could be, are there any number of limit, you know, trials?
No, I mean, actually the FDA process is pretty straightforward. So you submit, the only thing you have to be careful with is once you submit, they're going to start sending you letters back right away, and they're going to expect a response within a day or two. So if you don't respond to one of the deadlines, that's it. So you need to just, whenever you submit, you have to be ready, because they'll start within 24 to 48 hours, you start getting correspondence via
So
email.
I think sort of in next week's project with quercetin, we can also include as a control astaxanthins, and we can find more details like pharmacodynamics and kinetics, their lethal dose and EC50s. After looking at the safety, you know, I think it will be a good idea. We can also present, try to submit it next week.
Definitely, we will look into, you know, the excretion patterns of the metabolites of these particular bioflavonoids in the controls, and that'll give us a better idea. We'll consider things like the polarity of the metabolites, and we'll be able to understand which will be the most, which will be the best approach. We'll be able to have a better idea.
Absolutely. Absolutely. Saurabh, do you have some more things to say? More other studies that we're working on?
Other studies that we're working on currently, we will be working on a study on Medrol, and as I mentioned, we'll be doing a comparison with plasmapheresis combined with Medrol, but we can also do a pure Medrol study. And generally, the inflammatory response is beneficial and important in controlling viral activity through the innate lymphoid cell activity. So in this Medrol study, we will definitely assess ILC2 activity. However, in the case of cytokine storm, Medrol may be beneficial. So there's some tipping point at which the inflammation is excessive. At that point, I believe Medrol will be beneficial. Prior to that, of course, we need ILC2 activity, we need innate immune activity, but there is a point in this when the patient, the COVID patient, essentially decompensates, and then the approach has to be different. It's a little bit like heart conditions. When you are dealing with compensated heart failure, your approach has to be different than decompensated heart failure. So in a patient with a normal inflammatory response, you don't necessarily want to give Medrol. However, after the patient effectively decompensates and goes into cytokine storm, then you have to. So we want to understand when exactly do we define someone as needing Medrol? When will it be necessary to begin inflammatory suppression, immunosuppression? When is it worth to?
We are doing that with Dr. Sorry to interrupt. That's been defined. We are doing that with Dr. Jaskin. After the 5th day in the hospital, 5 days from the onset of the symptoms, and they start to get worse. Between the fifth and the 10th day, it's when all that cytokine storm it's mounting and the immune dysregulation. So we have that defined, and it's part of the guidelines that we use actually.
I'm aware. I understand. I've looked through the protocol. I just want to refine the line a little bit. Right now, I believe I know there's a line. Of course, it's not arbitrary, but I just want to define it in terms of specific biomarker elevations. Yeah. And we can only do that by using a protein biomarker discovery tool. That way we can create you can say once the ferritin hits 550, based on this clinical trial data, we have shown that it is better to immunosuppress. Prior to that, we cannot immunosuppress because it's shown to have negative outcomes. So we can be the people who create the guideline.
I agree. Totally.
I have one suggestion in that. How about Medrol with tocilizumab? They both will have a very good just to see which one is effective, because what we want to do that cytokine storms, Medrol might just block all interleukins or NF-κB suppressions, okay, which generally cytokines will not produce. But tocilizumab is very specific to interleukin 6. When macrophage produce interleukin 1 beta, 6, and TNF alpha, we can actually target any of these individually. There are medications which generally block individually too.
Yeah, we could use anakinra for interleukin 1 inhibition.
Yes, or TNF inhibitors, but these are generally to control some of those inflammation in cytokine storms.
I think it's definitely worth using those as control groups, because then you're isolating the study to inflammatory suppression, whereas with Medrol, you're actually combining immunosuppression and
but sometimes you don't want to suppress, sorry to interrupt, sometimes you don't want to suppress all the inflammatory markers. Like you need a fever as a cardinal sign, because that generally helps to eliminate microbes. But interleukin 6, the main problem is when acute phase reactant goes high, fibrinogens, right? All of those ferritin, that generally clog up blood vessel, and all these complications of ARDS, DIC can produce, right? So targeting interleukin 6 only actually can stop severity compared to when you are blocking the entire cytokine production from the gene level with steroid like Medrol, right? So I think it is good blocking all cytokines versus only particular, which generally is there any beneficial role?
Yeah, I think we can assess what biomarkers change when we specifically inhibit one interleukin or one specific acute phase reactant like TNF. We can assess what happens to everything else, and that'll give us a very clear picture of what we're actually doing to the patients, what changes are occurring. We can assess what's happening. Is there a different response in administering a interleukin 1 or 6 inhibitor to a mild COVID patient or a moderate or someone who's on a vent? What actually happened chemically? What changed in their epigenetics? And what new proteins increased expression? because everything we do will have a effect on the transcriptional regulation. Everything will cause up and down regulation. It'll change the signal transduction activation pathways, and that will be expressed only in a way that is encoded by the protein percentage increases and decreases. And we can only read that if we take some serum from the patients and put it through Olink. So
I agree with you. I think, yeah, that will be very useful, knowing the biomarkers, because that just gives a better idea, especially on result on lab. I agree with you.
Yes, exactly.
So Dr. Yatskin is going to be at the 3 o'clock meeting today. So that would be a good opportunity to get him involved with that one. He could be the PI on that. He's the director of our pulmonary fellowship at ARC. So he's going to have 4 fellows start pulmonary with him July 1, who may be interested in even being part of this. So let's bring that up to him today.
I would love to have any of them on board, and I would be more than happy to learn from them, because I'm sure they have so much more. I know they have so much more experience than me. So I'm really glad to have the opportunity to work with them.
It'd be great. So that's perfect. Okay. What else do we have? What other projects do we have out there? I know we have others, but go ahead and I'll let you.
I can do the Xofluza actually.
Okay.
This is Kunal.
Yeah, Kunal will explain everything about Xofluza. I can just guide her a little bit with the IND process, but she's taken a big role, and I'm really glad that she's she's taken a strong initiative in this. So yeah, thank you so much for that.
Thanks, Saurabh. So for Xofluza, we actually have a couple of things that we could possibly be doing. So I've talked to Dr. Novera. She has some data, so we could do like a retrospective observation for both inpatient and outpatient. So our team actually did put together ideas as far as how we could start doing literature review and start writing on it. And I've sent it to Uzam, and Muniba in our group has also helped me create it. So we're actually going to post that today. And then we're also thinking of, as we had discussed with the Genentech folks when Dr. Marcos and Dr. Michelle were on that call, we're thinking of doing like a single arm prospective observation study with Xofluza, possibly adding azithromycin to it, just like their original trial. So we reached out to Genentech, the manufacturers, and we're just waiting to hear back from them. As far as the IND, Sora, we have like over 70 people in that group, so we will certainly get started. And I will take notes from you as far as your experience, but we have plenty of people with all these projects that you have going on. So I think we will be okay. Unless you want to do like a different Xofluza trial, definitely bring that up. But as far as Xofluza, I think we're good for now.
I just think that we can obviously incorporate a trial focusing on which biomarkers are elevated in PH patients who are given Xofluza. And we can just do 3 categories: mild, moderate, severe, very simply. And that'll let us assess what actually happened to the patient from a biochemical level. And that can be done in
so if you want to write like a 1-page study rationale for it and just like have it ready, so we can present it whenever we have our next meeting with Genentech.
Yeah, of course. When is the next meeting?
So we don't have one scheduled just for yet. So as far as for all of these trials, not just for Xofluza trial, designing a trial would be one thing, but then coming up with a rationale that makes sense, like the manufacturers, like we have to give them some sort of buy-in and like come up with like a study rationale that makes sense. So I think what you're proposing, at least in my opinion, like keep that as a separate trial so we can get at least one trial going.
Jordan is also working at a pharmaceutical company, and he has some connections with people who can provide drug sponsorship for Larkin. So I believe that he's going to look into that for quercetin, and if needed, he can help us find someone who can provide recombinant BCG for the second BCG trial. He can provide someone who can find simple BCG, a non-recombinant version like the Tice strain from Merck. He can assess that. and he can also look into finding someone who will have tocilizumab access, as well as any kind of, I know we have a lot of Xofluza on board already, but just in case we need more, he can also help Kunal with that.
Sure. Yeah, and like we discussed earlier today with Merck and BCG, like let's definitely do some more homework, ask lots of questions, because when I hear quality, safety, efficacy, patient safety comes first. So let's at least ask questions. I have contacts. I will reach them out. We'll schedule a meeting, and let's make a list of lot of questions that needs to be answered.
I agree. Definitely have to do that. It's fundamental to getting set up for a sponsorship.
I agree with you. Thank you. Thank you for just coming on the board and giving me an opportunity to present this. What I will be doing, Saurabh, I will be sending out all the links, all those new links, the recombinant vaccinations efficacy, which are done in human trials or in mice trials. And I'll be sending those links in our group, so the all members, they can just go through it, and maybe it helps edit just to understand. What else do you want me to do on this project?
For BCG, I think the first most important thing is we get a PI. After that, we'll be able to understand what the PI is comfortable with. Because whether we're going to do a single blind or double blind study, that has to be determined by the PI and what they're comfortable with. So first most important thing is understanding who we will be working with, and we have to design the trial accordingly based with what they're comfortable with.
Sounds great. I agree with you.
Kunal, I have a question for you. This is Rana.
Hey, Rana.
How are you, Kunal?
I'm good. How are you?
Good, thank you. So, Kunal, while you're taking care of the pharma company part, what are we supposed to do in the group? As in, I have my literature search, but just asking by the next Monday, what should be my goal?
That's a great question. So we actually, Uzam, myself, and Muniba we're almost finalizing the literature review outline. So we're going to post that today. In the meantime, I know Dr. Adarsh is also working on figuring out how we can logistically have access to past data. But while we're waiting on that, we will certainly have the outline for the publication today.
Okay, thank you, Kunal.
Thank you.
Okay, Saurabh, so what's the next project?
So after that, we can look into more molecular approaches based on current mechanisms, and that'll help us evaluate the best targets. Because I've understood the approach for sending in INDs. I understand which clinical trials are commonly most effective, and there's some kind of pattern in these clinical trials, and I've come to realize that after reading so many. So based on the biomarker data that we get, after we understand a little bit about which biomarkers are actually elevated, we will be able to better choose new drug targets, new possible INDs. So basically this will have a snowball effect.
And Aaron, actually, do you want to say a little bit about our molecular lab capabilities so they're aware of what we can do in-house?
Yes, sorry. How you doing, everybody? It's Aaron here. So, yeah, in terms of next generation sequencing, I work very closely with Arpit and Joseph. We run all of the COVID testing via PCR. We currently have an ion from Thermo Fisher. It's a Gene Studio S5 Prime with a maximum read count of about 135 million reads on a S550 chip. The only thing is that it is costly. I don't think we'd be able to achieve around 300 per sample. A lot of these companies that do the full exome sequencing at around $300 to $400 a sample. The data typically ends up being resold to pharmaceutical companies and so on. But I think ARPIT can elaborate a little bit more on the sequencing capabilities in-house and what that would entail and look like.
Yeah,
I
so
want to mention that. I mean, I don't know if this cost is a significant factor, but in the case it is, we can always apply for an NIH grant.
yeah, thanks, Saurabh. I think this is important that we put together a grant proposal. But when we are doing the clinical trials, I think we have to keep in some other things in mind. That it's not just like the whole genome or whole exome sequencing. We should look at RNA-seq as well. That is whole transcriptomics, because most of the signatures which you guys are looking for as the drug is administered, there would be immune response which will be coming into the picture, and a lot of immune response pathways and genes. We should be looking post and pre-administration of the drug to compare whether the expression, the differential expression is higher, if a gene is upregulated or not, which will establish the biological mechanism of how this drug is actually influencing the immune response. And that will be a very good pilot study for us to go for a bigger NIH grant. So we do have the capabilities to do RNA-seq as well in-house. We can obviously look at like genetic markers, and then if there is a predisposition to a particular, a particular drug in terms of response. We can also look at sequencing the COVID genome, because there are a lot of strains which are coming in, and then given the amount of patients, positive patients and samples we have, we can easily establish if there is a particular strain which is affecting our local population. So considering all of those factors, that will give you insight into determining the epitopes which are specifically being present in the local population. So we do have the capability, but yeah, the cost is going to be an issue. If we can land a grant from NIH, that's going to be perfect for us. Because overall, I think once we have the trial or a positive result coming from the trial, the second question they will ask is the mechanism, how it has happened. And that will be important for any kind of FDA approval. We go further, it will establish us as an institute which has capabilities to do end-to-end approvals for the FDA going forward.
Keep in mind that genome-wide sequencing and genome-wide epigenetics is completely different. For genome-wide epigenetic, you need bisulfite sequencing because you are targeting the
No,
DNA
so bisulfite, I agree, Dr. Felix. So genome-wide would be...
Yeah, but
like
give me 1 second to clarify, give you a little bit of...
Go ahead.
So you're going to identify the protein biomarker. You don't need to do the transcriptomic because it's redundant information that you're going to have. So by the protein biomarker, that will be enough.
Well, sir,
So
how do you know that those are the only biomarkers that are going to be identified unless you do a full...
Because you have to do a shotgun. You have to do a shotgun. You cannot measure 10 biomarkers. You have to measure more than 100 biomarkers.
Wouldn't you want to have a second means to be able to establish that? Or would you trust...
That would depend. So that has to be considered based on what does the drug that you guys want to implement want to silence. If it is the protein, the secreted protein, so measuring the transcriptome is not needed. If you want to inhibit the mechanism, then yes, you need the transcriptome information.
Yeah, and then because we are doing it, in terms of already the clinical trials are there and the patients are present, if you're able to, add the transcriptomic information to the overall, project, also in terms of like consent, we would have to have genomic consent from the patient added to the IRB to do that. But it will be a very impactful overall project, given that if we cover all the bases in terms of like RNA-seq, protein-based, and also, the trial itself.
Right, but consider feasibility. So which approach is more feasible to implement in a shorter time frame?
Okay, I just heard that.
I think the turnaround times are not that bad overall for the sequencing itself, as you consider, like given that, we have the sequencer on board and that the lab is, pretty close to us. When we always prioritize, the samples coming from the clinical trial, we would be able to give a good turnaround time.
Yeah, I understand that point, but you have to be very cautious with the genetic data. You cannot measure the genetic data in batches. because it will create bias, batch bias, and then the data very difficult to interpret.
So we do have like, great experience, almost like 12 years running the bioinformatics pipelines of genomic data. We have done almost more than like 6,000 samples personally, not with, Cardiotropic. Of course, we are starting now. Personally, we have set up a very great, batch effect correction modules, normalization procedures, and QC procedures to kind of get rid of any kind of batch effects which are coming. Even at the low sample sizes, we are able to, generate great results with high accuracy.
Very difficult to generate power in genetic approaches with small sample size. So you have to be careful with that one.
Yeah, of course. Like if you're establishing overall...
the sequencers that we're running, I mean, it's all clinical grade sequence, semiconductor sequencing. So, I mean, the technology has advanced significantly over the past 5 years, and we have the latest model. And the software has definitely come a long ways.
Mm-hmm. Dr. Banks and Dr. Mehta, I would love to please have your contact information so we can discuss this in more detail and I can assess where we stand and how we can go forward. And I would love to learn more from you.
Absolutely, sir. Yeah, we'll share that.
Can you guys, can you put in yours and Aaron's information on the chat so Sarah can get it from there?
Yeah, right away.
Yeah, I actually agree with everything they've said, and everything which I've been speaking about biomarkers is actually under the umbrella of transcriptomics anyway. So they're right on the ball.
Let me suggest something here. For each study, everyone agrees that we have a one-pager rationale with a brief description of the summary of the trial. That's one thing. The other thing that we're not talking is what is the estimate amount of patients needed in each trial that needs to be included in that little page, and the estimate cost per patient for that trial. Because we're putting so many concepts together that is fine, but at the same time we need to have some kind of north and rationale, mini design, patient number, and associated costs. That will give us a better idea on where to start and what trials can be completed faster time, and we can have results for those specific trials in a faster time. So the team lead should be able to put the group together and basically come up with that. That can be shared in a Google Drive. I'll be more than glad to go over those to see if they make sense, at least operationally, to give us a starting point and a map, because I see too many moving parts, and when that happens, nothing gets accomplished. That's my opinion. So I think that's the very first thing is a group, a summary. That's what else.
Suggestion, Marcos. Definitely. I mean, I think this group can put it together. I think each of these projects is kind of running independently of each other. So I know it sounds like a lot, but they've got it pretty well organized. I mean, over this weekend, I got like 20 or so city certificates which over the weekend they completed both trainings. So I think we got a very entrepreneurial group that is going to make this happen. So I think he's right about we should have a one-pager, try to keep it as as straightforward as possible, and maybe break it down into separate instead of making one ambitious project, just break it down into smaller projects that then become more ambitious as we get more findings. But really, the sky's the limit as far as whatever you guys want to do. There's no limit, Madhu, at all. So you guys have an idea, we'll put it together as long as we have the patients to test it. And the good thing is between Cardiotropic and BioReference Labs, I mean, we have a big thousands of people that we've tested positive. So it's not like we don't have ability to do the trials. It's just a matter of organization and finding local people to help out. I think, Saurabh, I think we missed the ozone, right? Who's doing the ozone trial?
Yes, so I spoke with
Sanjay. Oh, sorry.
And after I spoke with Sheila, I talked to Dr. Richter also. And then some people from Team 11 had expressed interest in leading ozone. So Sanjay Mehet was the most interested in leading ozone. And he wanted to learn from me about what forms are used and everything. So I sent him a list of the approach that I took for the forms, and I helped him organize his group. And he's basically going to lead the whole team for ozone now. And he has acquired a Telegram team, and he has about 30 people in his group right now. And I've shown him how to assign sections of the IND for each part of ozone. So basically what they're looking at, I spoke with Sheila, and there was the team in Spain, and also a team in Italy. And I had talked with Sheila about possibly learning from both teams about the clinical trials that they did on ozone, so that we can avoid repeating any of their mistakes. We can do a Zoom meeting. I know that Sanjay would definitely want to be actively leading that Zoom meeting, and he will probably want to schedule it and these kind of things, so he can better understand what approach the Italian team and Spanish team took. There's also a IRB from Cleveland Clinic, and I believe there's a doctor there who gave approval for use of his IRB protocol in other hospitals. And I think Dr. Richter had expressed tremendous interest in leading ozone, and he was also interested in some bioflavonoids as well. And ozone, he has enough experience that it wouldn't be worth mentioning for me, because he has 10 years of experience with ozone. So I'm not sure if he's available at 3 o'clock, but
He will be. Yeah, he will be.
okay, then he will be able to better address the general approach that he wants to take in terms of control groups, experimental groups, that kind of thing. Because
yeah, I think his comments for he had some comments from what Sheila had sent as far as the IRB, and he wants to make some modifications. So let's just go with him, and Sanjay can put that together. They're connected, him and Sanjay.
Yes.
I know that Sanjay spoke with Dr. Richter. I gave Sanjay Dr. Richter's contact information, and Sanjay has sent Dr. Richter an invite on Telegram, and Sanjay has Dr. Richter's WhatsApp already.
Got it. Go ahead, Sanjay.
So we are actually we have just started. So I think Saurabh has explained everything already. So actually, I was in touch with Dr. Richter, and he did mention that for now we could just start with some reviews of literature, and we could go over something and have a plan that we could work on. So we're just in preliminary stages now. Hopefully, we'll come up with something good in the next few weeks.
Okay, perfect. Saurabh, did we miss anything, or is that pretty much everything?
The only thing remaining is the molecular approaches that we will design based on the current trials.
Perfect. And you got Aaron and Arpit available for any questions on that.
Yes, sir. Yes, sir.
I would like to clarify some of those statements, please.
Yes. Sheila.
The IRB I sent out was mistakenly a completed IRB, and my permission is in the historical information on it. So I would prefer people not be mentioning Cleveland Clinic, because that was information that was not necessarily, it was my mistake. Not a big deal, but let's just have clarity that that's not happening right now.
No worries. We want to be the only ones doing this, so we're good.
Well, we got to hurry, because I have a group out of Mexico that is very, and I've got another group out of California, and I'm supposed to be possibly working with a group out of Dallas, and I would love for Larkin to be first to the party, because you've been most amenable. Now, I did draft an IRB that has content from these renowned physicians in the industry that I'm currently waiting for their permission to see how they want their acknowledgments to be on the IRB. So these would be US physicians, and the protocols are in sync with those that have been used from Spain and Italy. to my knowledge of having done 1-hour interviews with both the Italians who require a translator, and I happen to have a PhD in medical translation for Italian. So that is something that kind of makes the communications a little more difficult, as well as time zone situations. And Dr. Hernandez does speak English, so that's a little easier. I do get prompt reply from both of those, Dr. Franzini and as well as Dr. Hernandez. They are both practicing physicians, so it's very difficult to coordinate meetings. So a lot of times, a lot of my interactions are off hours. I personally am available from 9 a.m. to 11. I am an individual. I don't have to, I do what I want to do when I can, how I can. So I'm very fluid. I look forward to working with you, Sanjay. What time is it right now where you are, Sanjay?
Hey Sheila, it's 10:30 p.m. right here in Nepal.
Okay, 10:30 p.m. Okay.
Sheila, just in regards to that, so Dr. Richter has a very specific idea of how he wants to put this together.
Right.
So he doesn't exactly want to follow what the Spanish and the Italians are doing.
Okay.
So he's been doing this for 20 years, like Saurabh said.
Fantastic.
So let's just do whichever way he wants to design it, because he's going to be the person on the ground. So
Now, Marcos writing the IRB? Did Marcos head that up?
no, well, Dr. Sanjay and Dr. Richter and that team is going to write submit to the IRB, and then the Marcos is going to take it from there.
Oh, okay. Sanjay, if you will give me your email, I'll send you a draft. It's just something to start from. Use it or not use it, whatever your pleasure.
Sanjay, did you want to make a comment?
Yes, Dr. Michelle. So I did talk with Dr. Richter, and the thing is that what he wants to develop is he wants to do auto chemotherapy for ozone on COVID-19 patients. So that's his actual plan. And what he wanted everyone else to do is to go over articles so that we know what auto chemotherapy is, and we could basically give ideas about that so that we could work on it.
That kind of broke up. I didn't hear that clearly. Can you repeat that, Jack?
I'm sorry. I do not have a very good connection. I'm sorry about that.
We can hear you now. Go ahead. Say it again. Repeat it again.
So the thing is, Dr. Richter was talking about auto chemotherapy.
Yes.
And he was talking about auto chemotherapy and how we could use these in COVID patients. And for now, he said that we could go over articles so that we could review about this and basically talk about this. So that's the plan for now. Auto chemotherapy.
Sure.
Okay.
That's
which is pretty much what we've been talking about all along, Sheila. So I think there's just some details that he wants. Which
is there any? My perplexity is what would be considered conventional care. Now, earlier, Saurabh was talking about the potential of kind of doing both testing. So I didn't know if maybe the conventional care could be considered somewhat palliative with vitamin C, vitamin D that everybody gets, and the experiment would be the MAH for ozone. Would that be possible?
Well, let's Sanjay and Dr. Richter look at that, and then they'll figure that out in their protocol. I mean, they're not done yet, so let's just look at. Aaron,
hey,
you want to introduce someone from Thermo?
Yeah, Dr. Michelle. So I have Nicolette Dutkin. She's with Thermo Fisher, the clinical application scientist. She specializes in next-gen sequencing. So now that she's on the line, I think we can bounce some of the questions off of her in regards to some of the cross-reactivities and stuff like that.
But before we do that, could I introduce Dr. Tom Schneider? He is a lung specialist in Jacksonville, Florida. Tom, are you on?
I see him on. Hi, doctor. He's muted, but
yes, I'm here.
Hi, doc.
Hi, I'm a clinician. I'm not a researcher, but I'm very interested in ozone. I'm impressed by the reports from Europe, Italy, Spain, etc., on how effective it's been at getting these patients better. So,
absolutely. Absolutely. We're going to work towards that.
So Tom, with their comments on being so open to, in some of the chat, they're talking about even sole practitioners on their quercetin. So Rob, I'm really all over that. I'm gonna get you some contacts with manufacturers. And it sounds like some of these great sole practitioners may be able to participate. So I'll be in contact. Would that be with you, Rob, or who would I do on the quercetin? Jordan? Okay.
Or did you mean for ozone?
Well, just I'm interested in the quercetin trial too.
Okay. So I will. I look forward to receiving what documents or publications or any approaches you have for quercetin, and I look forward to working with you. You have my email. Please feel free to speak at all about quercetin.
Thank you all.
Thank you. Aaron, go ahead, because we have 10 minutes before my next meeting. Do you want to go ahead and discuss with the thermal person on the line?
Yes. So what was the exact question in regards to some of the concerns around next-gen sequencing?
Felix.
So no, my concern was, or actually not my concern, my suggestion was that the epigenetic assessment is very expensive, even if you have the machine in-house. The reason is you're going to have a lot of targets that you need to measure, probably in the million targets. That creates a lot of experimental difficulty, including batch bias. So if you have 100 samples and you analyze 20 samples 5 times, so that creates a batch bias. And that is usually one of the limitations of epigenetic assessment. And combined to that, you're gonna have the cost, and if the pipeline of bioinformatic analysis is not carefully designed, the results may be false positives.
Nicolette, you wanna?
Yeah, so with this, the AmpliSeq panel that exists is designed to cover the full viral genome. So yes, that is a lot of targets. And we recommend starting with 1 million reads for each sample to get sufficient coverage of the virus, knowing that some samples are going to have a higher viral load than others. We're overestimating on the coverage side.
The epigenetic doesn't mean measuring the genome of the virus. It's actually measuring the genome of the host.
Oh, I'm sorry. I didn't realize we were talking about a different solution here. So this is you're looking at epigenetics. Okay, I thought we were talking about the existing COVID panel. So in that case, it comes from, you have to end up designing a new set of primers and also get sufficient coverage there.
Yeah, so in terms of epigenetic, thanks, Nicolette. Yeah, so in terms of that, casting a bigger net is always a good idea. Than going for like smaller targets. We would be lucky if we can discover a great biomarker, but eventually overall looking at the mechanism for epigenetics, it has to be a big blanket. And we have to look at all the mechanism. If we are going to publish in a reputed journal, the NGS-based epigenetic analysis is sought for.
That is I can tell you that is a misconception. You have to go from the outside to the inside. So you need to assess the protein shotgun first. And then from the biomarker that you identify in the protein, you reduce the target in the epigenetics. Otherwise, you will have too much information.
Yeah, but if we were to begin with the epigenetic with the RNA-seq, we already will have probably tapped all the potential biomarkers which might come out at a very low frequency as well. And the stability of the protein biomarkers is also in question. Like how, in terms of like the RNA sequencing or discovering the mRNAs, a lot of them might not even translate into, protein biomarker. It could be long non-coding RNAs, it could be microRNAs, it could be a lot of, the like circular, RNAs as well, which eventually will not translate into a protein-specific biomarker, but can be a potential biomarker for establishing the fact why this particular drug is working in terms of like immune response pathways.
So let me see if I understand. If you if you implement a drug, how are you going to assess on the patient that the biomarkers are getting reduced?
Of course, like,
RNA or protein?
No, immediate effect is, I agree with you with that.
It's a simple question.
But we are not doing it just for establishing the biomarker for the study. What we want to do is, increase, the potential, range of the study to have these kind of, follow-up coming up with RNA, and also with, the sequencing identification to figure out that whether,
that will bring a question of feasibility. They will bring a question of feasibility, and then you won't have additional lines of research. So you identify the problem first. The future directions of the research can direct then the mechanistic approach.
Yeah, but I mean, when we are establishing the study, we would have to think of these approaches to begin with. We can't just go later and say, hey, you know what, now we want to go back and sequence it. It would be better.
Arpit and Felix, why don't you guys form a separate group? And because obviously this is one of many things I consider it very important, but it is one of several projects we're doing. Might be the most important one, but maybe you can hash out this specific discussion separately. Would that be okay, Arpit? And
agreed. Sure.
This information, and also Joseph can be involved, Aaron can be involved, and Nicolette if she's available through her company. I do think we need to flesh these issues out.
I mean,
all right.
Yeah, yes, we can do that. No problem.
Yeah, I think that would be more productive. I don't want to, it's been 2 hours. I don't want to tie anybody up any longer. Saurabh, do you have any final closing thoughts?
I would just like to have her contact as well, along with Dr. Banks. I've received Dr. Banks and Dr. Mehta's contacts. The third one from their company, I don't have her contact.
Nicolette, can you put in your contact information? Is that okay? Your email, and then Felix, you can put your email address from Larkin University. If you can put it on
that's
the
fine.
chat. Yeah, and that way everybody can reach out and maybe try to have a conversation sometime this week so that this can move forward.
Yeah, definitely.
I mean, I love the discussion. Look, at the end of the day, this is science, right? Is people with finding reasons why to do something and why not to do something, and that's why we kind of need to consider everything before we launch it. But so I'm glad that we're having a lively discussion. It's great.
There is another thing, Dr. Michel, that I think epigenetic information that, we, if that trial goes, regardless of the cost thought, I think it will help even just knowing the host epigenetic information will help us in other trials too in BCG, even because the primary defense, our host immune cell, innate lymphocyte, macrophage, and dendritic cell epigenetic information, if we get that, it will give us a better idea what changes occur even after BCG. So I think in a long run, if it is, we still have to put more thought about epigenetic, either with Mehta, Dr. Mehta.
Yep.
So we'll I think it's a good idea, but I'll also agree with Dr. Felix that it could be, if we find the epigenetic information, it will be helping in other BCG trials.
Yeah, I'm just trying to read the devil advocate. The reviewers, the reviewers are going to target. That is the target that the reviewers are going to pay more attention will be the feasibility of the project. If the feasibility is shaky, it doesn't matter if the project looks like Miss Universe, it's not going to get funded. Doesn't matter how good it looks.
Makes sense. Well, I want to thank everybody for their time, and we'll see you next week at the same time. And in the meantime, we're communicating through the Telegram group. Whoever's not on the Telegram group, the information on the Telegram group is on the chat. I'll give you a couple more minutes to find the information that was just posted on the chat so that you can copy it. Thank you very much, everybody.
Thank you.
Thank you so much.
Nicolette, I'm gonna call you with ARPID, okay?
Sounds great.
Telegram ID somewhere? A Telegram ID?
Can somebody post the Telegram information for Sheila, please? If not, I'll give it to you later, Sheila. I can find it for you.
Thank you.
Welcome. Take care, everyone.
Thank you so much.
Thank you.
Thank you. Bye.